Okay, so welcome to lecture 25 already, and welcome back from reading break. It's nice that most of you made it here through the floods. So just the first note, just remember the exam is Monday, December 20th, 2 to 4. They have listed the space now in McKinnon Gym. There was some confusion about the A03 section, which is the section that's kind of a ghost section that only exists more or less on paper. It's the online section. All three sections are writing in the gym at this time. There is no online exam for the final. So I know there has been some strange information coming out from the university, but the official word is that all three sections, it's the same time, December 20th from 2 to 4. I don't know why we're the very last day, but it's unfortunate, but I guess you'll have lots of time to study, I suppose. So anyway, it's going to be, we're going to be using bubble sheets for it, so I will take one of the lectures and just sort of run through how we do that, because many of you might not have used the bubble sheets yet, because I know a lot of people are doing online exams, and I don't want you to sort of go in cold. So I will run through that, and I'll give you more details about it too. It's closed book, but you'll be allowed to bring in some materials that I'm still trying to sort that out, but it looks like you'll have a 2-page cheat sheet idea where you write your own 2-page notes. I just have to kind of figure out the best way to do that. So, is there any questions about the exam? All right, so we looked at the importance of desensitizing a signal system. The idea is we need to have an altering baseline, because what we care about, not we, ourselves, what they care about is differences in signal. Okay, is there more signal now than there was 5 minutes ago or less? And so there's a number of different mechanisms that can be used for that, feedback mechanisms, sequestration of receptors or internal targets, or degradation even of signals or target or receptors. We introduced a second signal mediator. We introduced phosphorylation. And so we have a signal that is, or we have a post-translational modification that's added, and that's in the form of a phosphate group. And it's added by an enzyme called a protein kinase. And usually that phosphate is transferred from ATP, but it could be other nucleoside triphosphates or even other molecules, depending on the system. But I think all the ones we're looking at, it comes from ATP. The reverse process, we're not putting this phosphoryl group back onto ADP to make ATP. Okay, that's not happening. Instead, this is added to a water, creating inorganic phosphate and restoring whatever the protein is to the original state. We don't like the term on and off. We're going to use un- or non-phosphorylated and phosphorylated in our terminology. And then on the right, we have monomeric GTPase, which we've seen lots of times. So looking at this signaling pathway, what we can see is it's very modular. And so we kind of came back to that because now we have a better understanding of what things look like and pathways and how cells work. And I showed on this slide, which of course, you haven't done a lot of genetics at this point, so I'm not asking you to fully understand the genetics, but what we can see, and I think it's obvious here, is that in 1 or 2 steps, domains can genetically be swapped around. And so this is, from the point of view of evolution, has been a big benefit because it's allowed really complex pathways, such as the insulin pathway, to evolve. But as a researcher, we appreciate the value of domains because domains carry a function, and by understanding what domains are in a protein or a pathway can give us insight into how that pathway functions. Okay? When we're looking at signaling pathways, we have a receptor binding a ligand, and one receptor may bind to multiple ligands. Okay? And likewise, a single ligand, or signal molecule, may bind to more than one receptor. So how can we determine which receptor binds which signal? One of the ways we can do this is through experiment. We can determine the affinity of that signal for that receptor, so how well it binds. And we can measure this with a value, which is a constant value called the dissociation constant. And what that really tells us is the ratio of unbound to bound at equilibrium. Okay? And so if the KD is a very small number, it means a signal binds really well. If a KD is a very large number, it means that signal does not bind particularly well. Once we're inside the cell, there's an integration of the signals. Okay? So we have different things that can happen. One receptor may activate several pathways. Several receptors may converge on a single pathway, so there might be redundancy in that pathway. Or we can get crosstalk between pathways. And the sum of all of these interactions tells us what happens in the cell. And finally, often pathways are arranged into these cascades. This can allow for amplification of a signal, as we see here, where we start out with one signal molecule, and in the end, we free up. So this signal molecule, we'll look at this pathway today, is the fight or flight response, where we want to free up glucose from glycogen. And at the end of this pathway, we activate 1 million. So we add one signal, we activate 1 million molecules of the enzyme which breaks the glycogen down, and we free up 10 to the 8th glucose molecules. So this massive amplification, but this also allows for several points of regulation, which can also be important. So today we're going to look at, sorry, this thing is making lots of weird noise. Today we're going to look at G protein coupled receptors, the largest group of cell signaling receptors in humans. We're going to talk about the fight or flight pathway, and we're going to talk about an enzyme called protein kinase A. We may get into phospholipase C, but I don't think so. I think we've got lots to talk about with the first few topics there. Again, if you're interested in medicine or embryology or fertility, we have a social hour tomorrow at 6 o'clock. This is online, so I'm going to be talking with Lisa and James tomorrow, and they're going to tell you sort of their different paths. So James is a traditional doctor. He's gone through med school, specialization in obstetrics, gynecology, and fertility. And Elisa, she was a student here at UVic, one of my first students. And she trained to become an embryologist, which is a different aspect, a technical aspect of the process. And so it should be fun hearing their stories. And if there's no questions, we'll talk about G protein coupled receptors. So there's kind of been two important Nobel Prizes given out for this work. We have Lefkowitz and Kabilka, and they were looking at the G protein coupled receptors. And the name may give it away, but the second Nobel Prize went to the actual discovery of the G proteins. So there's a G protein and a G protein coupled receptor. And in fact, the Nobel Prize for the G proteins was given before that of the G protein coupled receptor. So we're going to start by talking about the receptors. So the receptors look like this. So in this example, we have so they are 7 transmembrane pass-through proteins. So if we count this, they go through 1, 2, 3, 4, 5, 6, 7 times. If you go on to take biochemistry 300A, the textbook there talks about these as 7 transmembrane receptors. It's the only place in the world I've ever seen them referred to as that, but they do all pass through the membrane 7 times. So we can think about how they got embedded there utilizing the endoplasmic reticulum translocon. This means that there's a series of loops, okay, both in the extracytosolic as well as the cytosolic side. And it turns out that these loops are really important for the activity of the G protein coupled receptor. It doesn't look like this. It's not expanded like this in space. It's clustered, and we'll spend some time with the structure in a couple of minutes because that structure is important. We'll come back to this a bit later, but we can see in little yellow stars, it says these are sites of protein kinase A phosphorylation, and these ones in red say they are G protein coupled receptor kinase phosphorylation sites. So these are sites where post-translational modification of specific amino acids can occur. And it turns out these are regulatory sites, and phosphorylation of these sites leads to a downregulation of activity. Okay. So G protein coupled receptors, I have it here. This is the largest single family of receptors. There's more than 800 G protein coupled receptors in the human genome. Depending on what you read, anywhere from 25 to 50% of all drugs target G protein coupled receptors. Something to keep in mind with drug design is traditional methods of developing drugs. We didn't necessarily know the target, so we looked for compounds that would modify activities, so we looked for output, but didn't necessarily know the target. And so that's why this is an estimate. Most drugs that are designed now, you start with a target and you look for specific drugging of that target. And we can do this now because we understand pathways so much better and we have much better tools for analysis. Okay? But this is why when we talk about drug targets, often we don't really know what they are. So of the 800 G protein coupled receptors, about 390 of them are involved in a single process. Do we know what that process is? Does anyone want to guess? It's a sense, one of our 5 senses. Anyone know? You have a 1 in 5 chance of getting it right. Hey, that's just like a question on a midterm. So it's the sense of smell, your olfactory. Yeah. So almost half of our G protein coupled receptors are dedicated to one sense. And I'm going to introduce a word here that's important when we look at signaling pathways. They work what we call combinatorially. And what combinatorial means is that it doesn't mean we only smell 390 things. If we activate receptor A, B, and C, we smell one thing. If we activate A, B, and D, we smell a different thing. And so by working things in combination, what it means is you get a much higher number of possible signaling pathways because we can put them together in a huge number of combinations. All right? And so this allows us to smell a huge number of different smells. Ours are quite low compared to many animals. So for example, a mouse has over 1,000 olfactory receptors. So they have a much more extensive olfactory network. They can be arranged sort of phylogenetically, all right? And this isn't important for you to memorize, but they break down into 5 major groups. Okay? All of these major groups, though, they all consist of these 7 transmembrane receptors. So we have sort of 2 important studies. Actually, I'm going to start with this one because this was the first structure of a 7 transmembrane protein called bacterial rhodopsin. And so this was solved using X-ray crystallography in 1975 by Unwin and Henderson. And you'll learn in biochem courses that we can't just use a microscope to look at proteins and see their structure. We need to do different tricks and techniques. And one of them is to take a huge number of electron micrographs, so a huge number of photos of them from different angles and put them back together. And cryo-EM is now becoming a major technique for solving structures, but it's really only sort of in the last maybe 10 years, 5 years, where it's become really a dominant form of solving structures. One of the pioneers for this was Richard Henderson, and if we look here, this was solved in 1975. So this is a really early use of this technology. And one of the reasons that it's so effective is that if you have a protein that's embedded in a membrane, okay, a part that's embedded in a membrane, it's hard to solve the structure because you can't have it free in solution. Because we know if this goes free into solution, those hydrophobic components that are in the membrane are not going to behave properly, are they? And so these are some of the challenges of solving membrane proteins. We saw a workaround the other day when we looked at the receptor-ligand complex, and they just ignored the transmembrane part altogether and looked at the extracytosolic portion, which is all polar residues. So this is an astounding piece of work. And Richard Henderson shared in the Nobel Prize for development of cryo-EM techniques just 4 years ago. Unwin, unfortunately, did not share in the Nobel Prize, although Henderson and Unwin are still very good friends, and they're at the same institute, the MRC in Cambridge. The first crystal structure, so this is a much higher resolution diagram. If we look at this, we get the general shape, right? But there's not really a lot of information there. Okay? Remember that this was made before the era of computer modeling, so you had to physically construct your structure in three-dimensional space. This is a crystal structure, okay? And so this is two of the principal authors here shown that worked on this. One was in Japan and one was in Washington State at the time. And so they managed to solve the crystal structure of this, so much higher resolution. And this is rhodopsin. So this is the G protein coupled receptor we use for vision. And it's highly related to bacterial rhodopsin, this structure. And so this gained a lot of information. The first crystal structure of a non-rhodopsin came out in 2007, and this was Kobilka's work. And we remember that Kobilka was one of the people that shared in a Nobel Prize for their work on G protein coupled receptors. So the second component of this is the G protein. And unlike a monomeric GTPase, where it was a single protein, this is a heterotrimeric G protein, meaning it has 3 components that are different. There's an alpha subunit, a beta subunit, and a gamma subunit. Right? The alpha and gamma subunit both are lipidated, and so they are bound in the membrane. And this brings the complex to the membrane surface where it can interact with the G protein coupled receptor. Upon activation, it splits into an alpha subunit and a beta gamma. Okay? And really, it functions very much the same way as a monomeric GTPase. So the 2 types, of course, are the monomeric GTPase and the heterotrimeric. And the activation really works in an analogous system to what we see for the monomeric GTPases, the alpha subunit is bound to GTP, to GDP, and when it's bound to GDP, it associates with the beta gamma. There's a GAF, which is in fact the G protein coupled receptor. So the G protein coupled receptor kicks the GDP out, GTP comes in, we activate the heterotrimeric G protein, and then we deactivate it through hydrolysis of the GTP to GDP. And I'm going to show you the details of that here. So keep in mind the structure of the G protein coupled receptor is actually a cluster. And when we look at it, how these helices, these 7 helices, relate to each other is important for those loops in the cytoplasm. And when they bind to ligand, or in the case of rhodopsin, they're activated by light, those 7 helices, they change where they are with respect to each other, and that causes a physical change in the structure or the accessibility of those loop regions, and this allows them to act as a GTPA, as essentially a GAF. So we can see this here. We have a ligand binds to our G protein coupled receptor. Okay? This causes a change in the relationship between the helices, which allows the G protein coupled receptor to kick the GDP that's bound to the alpha subunit out. So GDP leaves, GTP binds. That causes a conformational change where now the alpha subunit separates from the beta and gamma. Both of these subunits, while membrane bound, have the potential to influence downstream processes. So they can both be involved in what we call downstream signaling. All downstream means is it's further along a timeline. So at this point, ligand binding might be the first thing, activation of the G protein coupled receptor would be second. Downstream of that, we have separation of the alpha beta. Downstream of that, the alpha beta can go do other things. So that's the idea of downstream. And we talked about that a little bit when we were looking at gene structure. So both of these can have downstream effects, but the alpha effects are better understood and more common, oddly enough. And so here we have our alpha subunit has broken away from the beta gamma. The alpha subunit in this example is interacting with this particular protein, okay? And what it does is it modulates the activity of this protein. Maybe it turns it on, maybe it turns it off, whatever it does, it's the downstream effect. With time, the alpha subunit will hydrolyze the GTP to GDP. This can be accelerated by a protein called an RGS, a regulator of G protein signaling. And really what this is it's the equivalent activity to a gap in the monomeric GTPase. So remember, the monomeric GTPases were capable of hydrolyzing the GTP to GDP, and we could speed that up with a gap. Here, the alpha subunit is capable of hydrolyzing GTP to GDP, but we can speed it up with an RGS. Okay. Once it's associated with GDP again, it reassociates with the beta gamma subunits, and now it's inactive. Okay. So really, it's pretty much the same as the monomeric GTPase pathway, all right, but it's a little bit more complex because now there's kind of a receptor and then a heterotrimeric GTPase. Any questions? Here is a list of some of the alpha subunits. There's an important note at the bottom here. There's 27 alpha subunits, 5 beta and 13 gamma. And again, these can work combinatorially. So they can be put together in different ways, therefore influencing different signaling pathways. And so from a small number of genes, we get a large number of combinations. So again, this important idea of the combinatorial nature of these. And it's a theme that actually comes up quite often in signaling, that many of these pathways use a small number of genes but combine them in different ways to increase the complexity of the pathways. So the main ones are sort of the G-alpha S, which stimulates something called adenylyl cyclase. We have other ones that inhibit. Okay, ALF is the olfactory, T is for vision, and the Q and O are for a pathway called phospholipase C. You don't need to memorize these outside of knowing what G-alpha S does, because G-alpha S we're going to spend quite a bit of time on in this class. Here's just some examples of the beta-gamma effectors. So this is just showing some of the pathways that they can regulate as well, just so you don't think I'm a liar, because anytime you pick up a textbook or you read about G-protein coupled receptors, they always talk about the alpha subunit. And so I just wanted to give you some proof that there are, in fact, beta-gamma influence pathways. I don't know what any of them are because I only ever teach the alphas, but there you go. So let's look at an example. This is a very well-studied pathway, and one that we often use as an example because it also leads into some pretty cool phosphorylation, and it's a really important pathway. So the fight or flight pathway, there's a number of downstream effects, but one of the main ones is this is the thing we were looking at the other day that leads to the release of glucose from glycogen. Okay, so glycogen, remember, is people's starch. It's a whole bunch of glucose molecules stuck together, and we have to free those up in the case of fight or flight so that the cells have energy for your muscles to either run away or fight. This same pathway is mediated through another type of receptor called a glucagon receptor. And what a glucagon receptor does is it responds to signals of low blood sugar. And so when we look at the pathway, the two pathways for the fight or flight or the glucagon are the same, downstream, but different receptors and different ligands binding to activate them, which is pretty cool. This is our first example of a secondary messenger. Remember, the signal doesn't come into the cell, but we can build a little signal molecule inside the cell. And in this case, we build a little signal molecule called cyclic AMP. And so this is built from ATP. So ATP at this point would have 3 phosphoryl groups, but what happens is it cyclizes on itself, and 2 of the phosphoryl groups are lost. Okay? And so this is called cyclic AMP. And cyclic AMP is it's an internal signal molecule, but most of its effects are actually mediated by a protein kinase called protein kinase A, which was the first protein kinase that was studied extensively. So what we have, and this particular figure is showing, because they are the same pathway, it's either epinephrine or glucagon. So epinephrine is the fight or flight signal, and it binds to an epinephrine what's called a beta adrenergic receptor. Okay, and glucagon binds to a glucagon receptor, but what happens downstream is the same. The alpha subunit, which is alpha S, that's why I said this is the important one to learn, activates a complex called adenylate cyclase, or adenylyl. That's not adenylyl cyclase, which produces cyclic AMP from ATP. So there's the overview from the textbook, and the textbook I don't like it doesn't really represent the receptor. It looks more like receptors we'll look at later, called tyrosine kinase receptors, but make a note that this is a G protein coupled receptor. They've just done a terrible job of drawing it. We bind to glucagon if it's a glucagon receptor, or epinephrine if it's a beta-adrenergic receptor, and we exchange the GDP for GTP in that alpha S subunit, which then goes on to activate adenylyl cyclase. Okay? The alpha subunit will then hydrolyze the GTP to GDP, and we break down, or we then reform the heterotrimeric G protein in its off state. Adenylyl cyclase is no longer in an active state. We can imagine this has to be very tightly regulated, okay? Because this regulation of blood sugar needs to be tightly controlled. It has a lot of metabolic effects. The cyclic AMP itself is broken down pretty quickly by an enzyme called phosphodiesterase, or PDE. And so PDE will break cyclic AMP down to just normal linear AMP, which can then be through steps we're not talking about, can be converted back to ATP. Okay? And the system is reset. Here's an interesting example of bacteria taking advantage of this. And so Vibrio cholera is a bacterium that causes the disease cholera. Cholera basically causes horrendous amounts of diarrhea where you have so much diarrhea you die because you dehydrate. And so very nasty disorder. It's associated with a lack of sanitation. And so from time to time there are outbreaks, and from time to time there have actually been sort of pandemic-sized outbreaks. Where we tend to see it now is often following things like a natural disaster or a breakdown of things like sewage treatment, whatever it might be. So these nasty little Vibrio cholera, they produce what's called an exotoxin. And this exotoxin is called choleragen. And what this does, and this is the part that's really cool from a 225 point of view, is they actually utilize endocytosis. So it binds to specific gangliosides, GM1, remember those are glycolipids, on the surface of a cell. It then induces endocytosis, and we get retrograde through the Golgi all the way back into the ER. So where this toxin needs to be, it needs to be inside the cytosol. How does it get out of the ER? It utilizes what's called the endoplasmic reticulum associated degradation. And what that pathway is it's a pathway that takes proteins in the ER that are misfolded, okay, and through a ubiquitin proteasome dependent pathway, what it does is it pulls those proteins out of the ER and disposes of them via the proteasome. If you think way back to our second section, we talked about this pathway. So what it normally does is takes misfolded proteins, pulls them out of the ER, and causes them to degrade. The exotoxin, what it does is it tricks the ER into pulling it out, but once it comes out, it doesn't go to the proteasome. It doesn't get degraded. Instead, it activates adenylyl cyclase, causes a huge ramping up of cyclic AMP, and one of the side effects of this is it opens up what's called this we talked about the cystic fibrosis transmembrane conductance regulator, dumps a whole bunch of chloride ions into the gut. We remember how this worked in the lungs. The body will flood sodium in to counteract the negative charge, and then we'll flood water in to counteract the increase in salt. And so this results in the massive amounts of diarrhea. But it's a neat combination of several things we've learned: the endomembrane system, protein degradation, as well as adenylyl cyclase activation. And the final one is that we look again at this transporter CFTR. So it pulls a lot of aspects together for us. So, okay. So the cyclic AMP then, what it does is it activates a protein called protein kinase A. So protein kinase A, in its inactive form, it exists in this tetrameric complex. So a tetramer has 4 components. They're actually all individual polypeptides. So it's actually 4 individual proteins associated together. The regulatory subunits, so the ones listed here as reg, these are a dimer. They exist as a dimer. And the catalytic components labeled cat are monomeric. And what happens is, in the absence of cyclic AMP, and this might be better illustrated in this slide, in the absence of cyclic AMP, this dimer, so this is the dimer, okay, regulatory dimer, binds into the active site of the catalytic subunit. And one subunit of the dimer binds to one catalytic subunit, the other binds to the second. Upon binding to cyclic AMP, the structure, which binds in these binding sites, the structure of the regulatory subunit alters so it can no longer bind into the active site. And that causes it to fall out of the active site, freeing the catalytic domains to mediate downstream effects. And most of the downstream effects in this pathway are due to phosphorylation by protein kinase A. This is showing the cyclic AMP bound versus unbound form. And so we can see there's a major change in the structure here that prohibits it from binding into the catalytic subunit when it's in the cyclic AMP bound form. So the cyclic AMP bound form is shown here on the left. And there's our molecule there. Okay, so this is showing the catalytic structure. This is a very interesting protein kinase in that it's a single domain. It's actually just the kinase domain. If we were to talk about the system as being modular, this is an example where the regulatory modules are actually separate proteins rather than being a domain within the protein kinase A. So this is, I guess, the simplest of the protein kinase domains, and it was the first one whose structure was solved. Okay. And so this was solved by the Korean lab. This is showing the structure, and all protein kinase structures have a similar look to this. Again, they'll recognize phosphorylation sites in a slightly different context. Some of them are specific for serines and threonines, as we have with protein kinase A. Some are specific for tyrosine, but they look the same. Okay? There's some slight differences in the structure of the domain, which gives rise to the differences in the substrates they actually phosphorylate. You don't need to, there's nothing really important for us to memorize about the structure in this course here. So phosphorylation is very rapid, and it can lead to those short-term, very quick alterations in activities of different protein targets. Okay? And protein kinase A is heavily involved in the regulation of glucose metabolism. And we spend quite a bit of time on that in 299 or Biochem 300b. Right? And one of the targets, remember, of protein kinase A is it can phosphorylate and downregulate the activity of the G protein coupled receptor. The G protein coupled receptor can also be phosphorylated by that GRK protein, the G protein coupled receptor kinase. And that creates sites for a protein called arrestin to bind. And arrestin will bind to the receptor and prevent downstream signaling. Okay? So we do have ways to regulate the pathway, a couple of different ways. But we can have short-term effects of protein kinase A, but we can also see long-term effects, and we can see changes in gene expression. And how that works is shown here. What we have is we have the cyclic AMP responsive element. Okay? And what was noted was that there were a whole host of genes whose regulation seemed to be linked to cyclic AMP levels. So if cyclic AMP was high, this gene would either be up or down regulated, whatever it might be. And so remember, we didn't have great sequencing technology and it was harder to identify proteins, but they did look at the promoter regions, and what they found was that proteins that were sensitive to cyclic AMP levels had a specific sequence, this cyclic AMP response element. And it turns out that cyclic AMP doesn't actually bind directly to that. It's bound by a cyclic AMP response element binding protein. Okay? And so that's called CREB. So this is a cyclic AMP response element binding protein. Okay? And this protein, CREB, doesn't actually activate transcription at this point. It has to be phosphorylated by protein kinase A. And when it's phosphorylated by protein kinase A, it's bound by a second protein called CBP, which is the cyclic AMP response element binding protein. It just rolls off the tongue. And so it can only bind to so this is why we use CREB and CBP. So CBP only binds to the phosphorylated form. Remember, this is only phosphorylated when we have active protein kinase A. And CBP will recruit RNA polymerase, which starts transcription. So that can lead to more long-term effects as a result of protein kinase A signaling. Is there any questions about that? Yes?
What exactly do you mean by short-term versus long-term effects? Is that just how long the signal lasts?
Yeah, so that's how long the signal lasts. I think a couple of lectures ago we had a slide where we showed some things are really quick in signaling. You phosphorylate a protein and alter its activity, or you, say, open an ion-gated channel. And so those effects might only last a couple of seconds in the cell. With changes in gene expression, they take longer to initiate because you have to make the RNA and the protein, but you might end up with long-term effects in the cell that could last hours, days, even more. Yeah, okay? All right, so I think we will end there. We'll see everybody tomorrow.